Hey, PBio, welcome to our discussion on chapter 2.1 through 2.3, talking about chemistry. So, starting off, some basic review. Everything around us is made up of matter. So, that's anything that has mass and takes up space. And when you start to break down the things around us into their most basic building blocks, you get an element or a substance that can't be further broken down into other substances. There's about 120 different types of elements, but only 92 of those will occur naturally. The others were synthetically made in a lab. And those elements are, as basic building blocks, can be put together to form something called a compound. Now, compounds are going to show us for the first time something called emergent properties. So, emergence is really important in biology. And if you remember one thing from chemistry, remember emergent properties. So, what emergence says is that as you go from one level of complexity to a higher level of complexity, there's going to be some new properties that we didn't see before or couldn't have guessed that they would happen. So, a great example is NaCl or sodium chloride or table salt. I'm sure you've all used salt to season your food before, and it's something you put in your body. But it's basic. If you break it down to its simpler components, those elements, sodium, which is explosive when it comes into contact with water, or chlorine, which is a really toxic gas that's bad for the human body, are both things you don't want inside you. But when you put them together, a new property forms. We have delicious table salt. In our body, we have a lot of elements, but there's four big ones that make up about 96% of our body. That's Big Chon, or CHON, carbon, hydrogen, oxygen, and nitrogen. You also have some minor elements that are in that remaining 4%. The majority of that remaining 4%, which is things like phosphorus and calcium that you'll see a lot as we talk about how cells communicate and interact and what goes into them. And then there's about 30 to 50 trace elements that you have really, really tiny amounts of in your body, like zinc and copper. All right. So, those elements, if we break them down to their simplest component that still has all the properties of that element, are called an atom. So, an atom, as you can see in this diagram, has protons and neutrons at its core, which are protons, positively charged little particles, and neutrons are negatively charged particles. And then they're surrounded by a cloud of negatively charged particles called electrons. I'm sorry, I misspoke. Neutrons are neutrally charged or have no charge. Elements, each element is differentiated. So one element is different from another based on how many protons they have in the nucleus. So all we care about is how many protons are in the nucleus to determine what element it is. Now, if electrons or neutrons change, we have different forms of that same atom. So, for example, an isotope is where you have more or less neutrons. Now, since neutrons have mass, that's going to change the total mass of that atom. So you may have heard of things like heavy water, which have an isotope form in it. And some are really stable and some are unstable. So I'll talk about what that means. But when you see things like C12, C13, that's saying how many neutrons are in the atom. So C12, 12 neutrons, C13, 13 neutrons, and so on. An ion is when you change the number of electrons. So a cation is when you have more protons than electrons, so it has a net positive charge. An anion is a negative net charge, which means you have more electrons than protons. And ions are going to be really things that really want to form bonds so they can get stable. So I mentioned unstable isotopes, and these are going to have something called radioactivity. So radioactive atoms are radioactive because they're going to spontaneously emit these high energy particles until it gets stable. And radiation can be really damaging to biological systems. It can alter DNA and cause all sorts of problems in our cells. However, we also like radioactivity in biology because it allows for us to scan for things we can't see with our eye or even with a microscope. So, in a famous experiment by Hershey and Chase, they proved that nucleic acids were the material of biological heredity by tagging little viruses with their proteins with an isotope, and then their nucleic acid or their DNA with an isotope, and then tracked how that moved through the reproductive process. They found that the radioactive isotopes only stayed when it was tagged on the DNA. So radioactivity is really useful for tracking things in cellular biology. All right, so looking at our atoms, it's important to understand how they're held together with energy. So energy is going to hold electrons close to the nucleus. It's going to be something called electronegativity. So more electronegativity is going to pull that electron a little bit closer. When electrons absorb energy, they can move to a higher energy state or move further from the nucleus. So the further an electron is from the nucleus, the more energy it has in it for going against that electronegativity energy, pulling them in. So if an electron does move closer in, it's releasing energy. It has less overall energy. And where it is in relation to the nucleus is going to be referred to as its electron shell. So electrons are stored in shells in groups of 8, except for the very first electron shell, which has 2. So it's 2, 8, 8, 8, 8, so on. Whatever the outermost shell, we refer to as its valence shell. And that's going to be really important for understanding bonding, is how many electrons are in the valence shell or the outermost shell of an atom. So chapter 2.3, we'll talk about that bonding. So quickly, we'll go through a couple different kinds. Covalent bonding, where electrons are shared. So here we have hydrogen with 1 electron. It needs 1 more to fill its valence shell. So instead of stealing 1, what it's going to do is find another hydrogen ion and share. So these 2 electrons between the 2 are going to pass between both atoms. So they each have a share of 2 electrons and can both fill their valence shell. This is going to allow them to be held really closely together and form that bond. Ionic bonds is where an electron actually gets transferred. So talking about sodium chloride again, sodium has 1 in its valence shell. Chlorine has 7 electrons in its valence shell. So it's a lot easier to just lose an electron than gain 7 electrons. So sodium is going to drop down in energy level. Chlorine is going to gain an electron to fully fill its valence shell. So now both of them have fully filled out valence shells. But because that positive-negative interaction, that's going to create a really strong attraction between these two and form a really strong bond. So we form bonds through something called a chemical reaction, which is where we either break or make a bond. And the big thing I want you to remember when we're breaking or making bonds is that mass is conserved. So you have to have the same amount of atoms on both sides of a reaction. It has to balance. Energy is conserved. It's not going to be made or lost. And charge is going to be conserved. So if we're at a net neutral over here, we have to be at a net neutral over here as well. A couple other types of bonds. We have covalent bonds that are polar. So when one atom in a covalent bond is a little bit more electronegative than the other, it's going to, even though it's sharing the electrons, it's going to pull those electrons on average a little bit closer to that atom than the other ones. It has more electronegativity. And so as a result, you have a atom that's a little bit more negative versus the other ones that are a little bit more positive. Now, depending on the shape, if you have one side that's more negative and one side that's more positive, so here you have like a dipole, you have a negative side and a positive side in your water, that's going to mean that it's polar, or it has a negative side and a positive side. And polarity is going to allow for something called hydrogen bonds. And these aren't true bonds, just really strong attractive forces. So we have a more positive hydrogen and a more negative oxygen. And there's going to be an attraction between them that's going to hold them close together called a hydrogen bond. We'll talk a lot about hydrogen bonds throughout biology. And why are we so obsessed with bonds in biology? I know this isn't a chemistry class. Well, if you remember one other thing besides emergence out of this whole chapter, it's that shape is really, really, really important. Shape is so important in biology. It determines if things can interact, and all the complex systems in our cells come down to, do 2 molecules have the appropriate shape to interact and cause a reaction? And shape in molecules is exclusively due to how things are stuck together and how it's bonded. And so just remember that bonds are going to determine shape, and shape determines if it can work in a biological system. All right, before you leave, make sure you can explain how elements are different from each other, the differences between ionic and covalent bonding, be able to talk about emergent properties, understand what a polar covalent bond is, talk about bonding and shape in biology, and be able to remember a use for radioactive isotopes in biology. All right, that's it. Talk to you next time.